Hello future doctors, कैसे हो आप सब? Welcome to today's high yield session. I'm your host and today we're tackling a massive, incredibly important topic for your exams, cellular messengers and receptors. Okay, let's just dive right into this. We're going to map out the entire journey of a cellular signal, literally right from the moment a hormone arrives at the cell membrane all the way to the complex machinery it triggers inside. आज से concept बिल्कुल clear करना है. So, grab your mental notebooks, because we are breaking this down step by step.
आज हम क्या कवर करने वाले हैं? First, hormone and receptor classification. Second, extracellular receptors and GPCRs. Third, second messengers in action. Fourth, kinases and intracellular receptors. And finally, some absolutely high-yield clinical pearls. चलो, शुरू करते हैं.
All right, Section 1, Hormone and Receptor Classification. Think of this as our VIP list.
Before we can really understand the receptors, हमें उन VIPs के बारे में पता होना चाहिए जो मैसेज डिलीवर करते हैं. Notice how simple biology categorizes these VIPs into four main classes. First, we have amino acid derivatives like tyrosine, which gives us catecholamines and thyroid hormones, and tryptophan, giving us serotonin and melatonin. Second, we have peptide and protein hormones, like the 51 amino acid insulin or the 84 amino acid parathormone. Now, ध्यान से सुनो, this is absolutely crucial. These first two classes, amino acid derivatives, except thyroid hormones of course, and peptides, cannot cross the cell membrane. They just can't. They need extracellular receptors. On the flip side, we have cholesterol derivatives, which are our steroid hormones and vitamins A and D. Because these are lipid-soluble, they get the ultimate VIP access. They cross the cell membrane directly and bind to intracellular receptors. ये concept हमेशा याद रखना.
Actually, before moving forward, let's clear up a basic definition so you don't get confused later. The hormone itself, that is simply the first messenger. It's literally the postman knocking on the door. It binds to the receptor on the outside. The second messenger is the signal generated inside the cell, things like cyclic AMP or calcium. The first messenger never actually enters the cytoplasm in these cases, it just delegates the entire job to those second messengers. Samjay?
Moving on to Section 2, Extracellular Receptors and GPCRs, aka the bouncers.
Let's zoom in on the cell membrane surface and look at the most important bouncer of the cell, the G-protein-coupled receptor, or GPCR. This is a transmembrane protein, also famously known as the serpentine or 7-transmembrane receptor, because it weaves through the membrane exactly 7 times. Connected to it inside the cell are 3 subunits, alpha, beta, and gamma. Now mark this in your brain. This is a direct exam pearl. It is the alpha subunit that has the GT-pace activity, not beta, not gamma. Alpha is the one doing the heavy lifting here.
So the crucial point is how this mechanism actually fires. Let's walk through it slowly. Step 1, the hormone, our first messenger, binds to the GPCR on the outside. Step 2, the alpha subunit gets activated and actually physically separates from the beta and gamma subunits. Step 3, this alpha subunit uses its GT-pace activity, hydrolyzing GTP to GDP, which liberates energy. Step 4, this energy is then used to send our second messengers inside the cell to regulate all those downstream reactions. Your concept ekdam clear hona chahiye. It is literally the foundation of cellular physiology.
Okay, Section 3, Second Messengers in Action. The relay race begins.
So the postman is knocked, the bouncer has opened the door, and the relay race inside the cytoplasm begins with different flavors of G proteins. Memorizing this relationship is an absolute goldmine for MCQs. GS is stimulatory. It stimulates adenylyl cyclase and increases cyclic AMP. GI is inhibitory, so it does the exact opposite. It decreases CAMP. Then we have GQ, which is all about increasing intracellular calcium. And finally, GT, which acts by transducin in your retinal rods, activating PDE enzymes and actually decreasing cyclic GMP. Now let's see how this builds into actual physiological effects. Look at the contrast between CAMP and calcium as second messengers. Cyclic AMP handles metabolism like glycogenolysis, and it's the messenger for secretin to increase bicarbonate in pancreatic juice. Calcium, on the other hand, triggers uterine contraction via oxytocin and exocytosis for TRH and CCK. But the Oncedeco, here's the classic tricky pair for exams, vasopressin. Vasopressin V1 receptors use calcium to cause vasoconstriction, while vasopressin V2 receptors use CAMP to insert aquaporin 2 for water reabsorption in the kidneys. Same hormone, different receptors, and completely different second messengers.
All right, Section 4,
kinase, and intracellular receptors, the VIP axis.
We've talked a lot about GPCRs, but what about the alternative pathways? On the cell surface, we also have kinase receptors. Let's do a rapid-fire association here, because you really need these on your fingertips. Insulin, it works via the tyrosine kinase receptor, which is absolutely crucial for cell growth and differentiation. What about growth hormone, prolactin, erythropoietin, and leptin? They all use the Janus kinase receptor, specifically the JAK-STAT pathway. And serine kinase, that's used by reproductive hormones like activins and anti-Müllerian hormone.
Now let's talk about the hormones that actually enter the cell. Remember our steroid hormones and vitamins from earlier? They don't stop at the membrane. But please, inko mix up nahi karna hai. Some wait in the cytoplasm, while others go straight to the nucleus. Aldosterone, cortisol, testosterone, and vitamin D bind to cytoplasmic receptors. Meanwhile, thyroid hormone, vitamin A, estrogen, and progesterone head directly for the nuclear receptors. Let's quickly break down the structure of these nuclear receptors. The receptor has different domains labeled from A to F. The A/B domain near the N-terminal contains activation factors to actually start transcription. But focus right here. The C domain is the DNA binding domain. That's exactly where it attaches to the gene. And the E domain is the ligand binding domain. This is where the steroid hormone physically attaches to the receptor itself.
Finally, Section 5. High-yield clinical pearls. What happens when things go wrong?
This is where we apply everything we've just learned. What happens when this beautiful physiological signaling breaks down? Let's look at bacterial toxins. Cholera and pertussis toxins are notorious for hijacking our GPCRs. Cholera toxin continuously stimulates the GS subunit. The result? Pathologically high CAMP, causing massive electrolyte secretion, severe diarrhea, dehydration, and potentially even death. Pertussis toxin, on the other hand, inhibits the GI subunit. Remember, GI normally decreases CAMP. If you inhibit the inhibitor, you again get pathologically high CAMP. This leads to electrolyte secretion in sputum, causing whooping cough. Donotoxin CAMP increase karte, but through completely opposite mechanisms on the G protein subunits.
Okay, here are 2 absolute 5-star clinical points for you. If there is a gain-of-function mutation in the GS-alpha subunit itself, you get McCune-Albright syndrome, which is characterized by precocious puberty, skeletal malformations, and café au lait spots. Next, remember tyrosine kinase, which we said regulates cell growth? Well, in chronic myeloid leukemia, or CML, a BCR-ABL translocation activates tyrosine kinase pathologically. It literally just stays on, causing uncontrolled cell growth. The magic bullet treatment here? Imatinib mesylate, a fantastic drug that specifically blocks that tyrosine kinase receptor.
Let's briefly wrap up the neurotransmitter side. Receptors here fall into 2 different speeds. Ligand-gated receptors, like nicotinic acetylcholine or GABA-A receptors, activate ion channels directly. They give you incredibly fast responses. GPCRs, or metapotropic receptors, like muscarinic acetylcholine or GABA-B, have to act through all those second messengers we just discussed. Therefore, they give a much slower but more sustained response.
Before we wrap up, I want to leave you with this provocative thought about receptor homeostasis. Normally, as a safety mechanism for hormone overactivity, our bodies internalize, inactivate, or desensitize receptors, making them less responsive. Now, if a new drug could permanently halt that desensitization, keeping our receptors firing at 100% all the time, would we become superhumans, or would our cells just exhaust themselves to death?
Sochi iske baare mein.
Thank you so much for joining me on this session. Keep revising these pathways, literally draw them out on paper, and I'll see you